From here, I want to switch gear because I know David wanted me to talk about mind body and stuff, which actually I got very excited recently. We got a paper accepted a week ago, so I will be happy to show you some results. So using from this EEG or from the electrophysiology, from this hemodynamic response, one thing we can do is to understand the brain. But there's one more step. By understanding brain, we want to either you want to say aid the diagnosis or treatment of disorders. Another thing is you say, okay, can we help a patient or help those people and who need help to do something more? So that is really the scientific goal of something called brain-computer interface. This is a research field recently received quite a bit and attention. So you have a brain, you have a computer, and how to interface them together. Now this slide illustrates the general approach in this field, and there are various means of the brain-computer interface. You could use invasive recording, like for example, you can record the local field potential, a spike chain, or ECOG, or another way is you could record non-invasive EEG. Regardless of what kind of neural signal you're going to record, you can decode that signal, extract the information, and to control external and device, or external and action. You can modulate the environment. You can help a patient to do communication, such as surf the web, or you could turn on the TV, switching the channel, making phone call, or control robotic arm. You could control a wheelchair, and you can certainly fly a model helicopter. I will show you. Yes, we are able to fly a model helicopter. So this slide just illustrates one of the basic research we have done, that in order to accomplish this goal, especially from non-invasive EEG, we have been combining this source imaging approach and to better the decoding of the signal from brain-computer interface. In this case is motor imagination. It's amazing. Motor imagination sometimes being used in the mind-body awareness training, but in this case is really a more concrete. We ask a subject, say, okay, you move your left arm, and then next is you don't move. But you're just thinking what you just have been moving. And same thing, you imagining moving the right hand, but then you really don't move anything, because in real patient, you may not be able to move. And then that kind of motor imagination was decoded and extracted and as a control signal. I will show you here, actually, it's very clearly illustrated here. In the subject, if you do not do the motor imagination, you will observe a idling 10 hertz. It's called mu rhythm, or actually overlapping with alpha rhythm, because it's in the sensory motor cortex that we call the mu rhythm. In visual cortex, otherwise you sometimes call alpha rhythm. It's 10 hertz rhythm. You see that keep going. But once the subject starting imagining, you can see this disruption of such an idling 10 hertz and the rhythm. And such kind of phenomena, which is called something called event-related desynchronization, or ERD signal, that is one of the really neurophysiology and background for behind many of the brain-computer interface system in the world. And in this case, that actually we combine the source imaging, try to understand where such kind of ERD signal come from and why people can, generating such kind of signal control a device, because by understanding why, we can design a better system. So I think I'm not going to talk about this details. We did have to develop a new technology because there was no technology out there to do the source imaging of single trial. And because imagining, it's not like if give the visual stimuli and you average them, that is the visual evoked potential, which is very strong. But now you're thinking very subtle signal, so it was not able to image before, but we have to develop some technology. Here is just show you one example. We use this paradigm actually in our helicopter control. I'm going to show in a few minutes. So in this case is, if a subject, in this case, if you move this cursor to the left side, subject being asked to imagining moving left hand. And then we found that actually, we flip that left to go to right, right to go to left, as what we know is contralateral and motor activation. And you will see the source imaging of the motor cortex for the left movement, for the right movement, and going up is simultaneously both. And for going down is we ask the subject, intentionally imagining nothing. And basically it's a silent background. That is clearly being mapped using this EEG source imaging and technique in this case. And one more step as what we have taken to integrate EEG with the functional MRI, we did the same thing because this is really reflect the study design. In animal, you are always able to insert the microelectrode to do direct recording in the region of interest. So you can validate your result or you just study directly. But in human, that is really limited. And to my understanding, I mean, unless we use a totally different kind of independent measurement, and otherwise it's very challenging in healthy human subject how you can do it. But if we could get the same results from EEG, from fMRI, that actually provide really and complementary information can affirm or confirm our hypothesis. So in this case that we did this fMRI EEG co-localization. First of all, we started with the movement and the bold activation first. In this case, you can see a subject being asked to moving the left hand. You can see in the right and hemisphere motor cortex, the biggest bold elevation is being observed. Now, if the subject is moving the right hand, then left motor cortex, the biggest bold elevation. This is well known in the neurophysiology. Now, in the same subject, we ask subject, don't move. Now you're imagining moving. Very interestingly, that you observe this bold elevation in the contralateral motor cortex while you imagine moving instead of actually executing the movement test. Then here is a group of 13 human subjects. We look into both fMRI and EEG. You can see if you move the right hand and then left motor cortex, the bold can show the elevation and clearly activation. If you imagine moving the right hand, similarly, you see the bold activation, but in a much focal and area. And similar for EEG, from the beta rhythm, from the alpha rhythm, and contralateral motor cortex will be lighting up if you moving and or imagining. And here just showing imagining right hand, imagining left hand, imagining foot, and for the bold and for the EEG result. And one thing we learned, it's quite interesting that usually bold signal is going to be elevated if you imagining moving, but EEG signal, the power going down. Due to that mechanism we described, is there is idling activity if you do not imagining, but once you're imagining, you disrupt that, so the power goes down. But that different signal is proportional to that this multi-imagination activity.